to talk about an idea I have for a more accurate molecular or genetic clock. Genetic clocks are a way of predicting when stuff happened in evolution by looking at the rate of mutations and how many mutations occurred, and also how diverse a group is. The idea is that longer a group lives, the more diverse a group becomes, and the more mutations occur. Fine idea, but there's a lot of problems with it because mutation rates can be highly variable. Not only can they just be highly variable, you could have bottlenecks that could reduce diversity and get rid of a bunch of ones. You could have population explosions that let more survive. And there's also problems with sample size and stuff like that. However, what is a lot easier to do is first construct just a simple family tree that doesn't look at how far back stuff goes. Just like, are they brothers and sisters, uncles, aunts, or how distant relatives they are. Now, in this simple case, you can mostly ignore mutations this first round. Then you can look at, from this simple case, you can see how fast mutations occur. Also, molecular genetic clocks work less with time, more with actual how many generations. That's the thing. So if you have soil generations, so more accurate to tell you how many generations back, but that's usually converted time by some proxy. So now we look at how many generations are generated by how related, and then we can look at brother and sister and get the rate about how many mutations occur, about between how many mutations we see in a brother versus sisters, or how much we can attribute to that. We can do some fancy math, but sometimes we can just look at a gene and see how many is different in a population. It's not perfect, because if we don't have the parents, we don't know what the original DNA was. So we will have to do some guesswork there, but we can get kind of a sense. But not only that, another problem with genetic clocks is some mutations occur much more frequently than other ones. One mutation is you change one letter in the genomic sequences, but some letters are easier to change than other letters, because they're less or more stable. For example, I believe C and G are more stable than AT, because C and G's have more bonds in between them to stabilize them. Let me just look it up quickly. Nucleotides. And you can see my horrible spelling. But yeah, CG have 3 pairs where only AT have only 2. So the bonds are weaker between them, allowing for more ones. But sometimes there's other things like that are more common than you would think, like whole big chunks of DNA can be copied. So bigger changes can sometimes happen more easily than small changes. Also whole big sections can be deleted. Then whole big sections can switch around with chromosomes when chromosomes are switching genes and stuff like that. And other things can happen during the chromosomes doing stuff. A lot of it happens during crossover, when the DNA switches genes between other pieces of DNA during pregnancy. The formation of new cells. But there's also genes that can actually jump in your cell and duplicate. And then there can be big changes of like large events that causes rapid change, like a massive burst of radiation or something that hits a particular section really bad or cuts out a section. Then viruses can insert the genomes into stuff and it can get stuck there. So beyond what this method could also do is we could figure out how common big changes are, or different types of changes are, and we can start making a prediction system. Says in this organism, because it also varies from organism to organism, these type of changes occur more common in this one at this frequency. Then we can use all this to adapt a molecular clock to go more accurate backwards and figure out how fast different stuff changes by fitting this both. I mean, just iteratively. That means you try it over and over again, try different values and converge onto a good value. And we can use some machine learning to speed up process by finding patterns and figuring out what direction it's heading. Now, once we generate a basic tree going back, we're more confident about everyone's relationship and how distantly or closely related. Then we can fine-tune, better yet, the rate of mutations throughout this whole entire thing. By that I mean now, before we were only confident maybe 1 or 2 generations back about how much everyone is related to only to here. But let's say we make a new tree and there's some confidence going all the way. In this new tree we're somewhat confident all the way back up to here, because we learned about time and everything, and we learned about what's mutations and what's not in between stuff. So then, with a measure of uncertainty that we actually can compute better, we can use this to go back even more in time. Not perfect, but to some extent, even more in time. Then we can make this tree better, and so on, and keep going back and forth until it converges to the best we can do. There's always going to be imperfections with this method, but I think it's a good idea. Well, thank you very much for listening. I hope you found this interesting. This could also serve as uses to understand particularly how viruses are mutating and spreading, or other diseases. It could also help us better understand how evolution happens, like how common different types of changes are and how detrimental they are, which could lead to us designing better ways of speeding up evolution to create better breeds of plants or bacteria for cell cultures or everything. However, I don't know until we start experimenting how well this can get. Please thank you very much again, and please like and subscribe. It really helps the channel out. If any idea for future videos or comments, please leave them in the comment section below. Thank you very much. Goodbye.